Hello students, welcome back to the biology class. So in the last class we started the topic of nucleic acid. We understood that it is a linear heteropolymer and we understood that it is called as a heteropolymer. The reason behind is the difference in the nitrogen bases, because on a piece of DNA or a piece of RNA it will never be the same nitrogen base repeated. Naturally it would have random arrangement of A, G, T, C in the case of the DNA and A, G, C, U in the case of the RNA. Therefore, it is called as a heteropolymer. The sugar molecule and the phosphate group, or you can say ribose sugar and phosphate group, would be same in the whole RNA, and deoxyribose sugar and the phosphate group would be same in the whole DNA. So the variable portion is the nitrogen bases. It is a linear arrangement, so that's why it is called as linear heteropolymer. We learned that it is also called as polyribonucleotide or polydeoxyribonucleotides according to whether it is made up of ribonucleotides or deoxyribonucleotides. We understood in the last class that it was first isolated by Friedrich Mischer and he called it as nuclein, and later because it was discovered that the DNA and RNA, they are acidic in nature, so this acidic property contributed to the name to be changed by Altman and he called it as nucleic acid. We learned in the last class that the nucleic acids are present in all living organisms including the eukaryotes, prokaryotes as well as the viruses. And when we talk about the DNA and RNA, both are present in the prokaryotes as well as eukaryotes, but viruses would carry either DNA or would carry RNA, will not carry both of them. We even learned that we learned in the last class that the RNA is the ribonucleic acid and it is unstable, it is reactive, it came earlier in the course of evolution, and we learned it is single stranded, but there are exceptional organisms where double stranded RNA is also present. So universal thing is what, that the ribonucleic acid is supposed to be single stranded. It is made up of ribose sugar, the reactive one, because of the OH group present at the side in the side chain of the second carbon in the furanose sugar molecule, and then it carries uracil, cytosine, adenine and guanine. And we learned that the RNA formation occurs in the nucleus, so it is done with the help of the DNA, and this DNA will cause the formation of the RNA by the process called as transcription, and then it will start functioning as various types of RNA. So we started in the last class the types of RNA and we covered all the easier types of the RNA, that is the catalytic one. Catalytic one is the one which is reactive in nature. So in 11th we have learned that the RNAs can be enzymatic in nature. So there are some enzymes which are made up of RNA. Example ribozyme, example ribonuclease P, example peptidyl transferase, we'll be learning about later. So these enzymes, and then we have microRNAs which are regulators. They join to the gene to be expressed and increase the expression or decrease the expression, so they regulate the process of gene expression. Then we learned in the last class hnRNA, that is the heterogeneous nuclear RNA, which undergoes splicing, capping, tailing, which will be covered in this chapter later, to form the functional mRNA. hnRNA is found only in the case of eukaryotes, and we learned this name was given by the Winberg, and according to Winberg, all those RNAs which are lesser than 8S in their sedimentation coefficient value are called as heterogeneous nuclear. snRNA is this small nuclear RNAs which join with the proteins to form spliceosome or splicing bodies, which help in the process of splicing, which turns hnRNA into mRNA. We learned even in the last class that there is scRNA, that is the small cytoplasmic RNA which joins with special protein to form signal recognizing particles, so that the ribosomes joins to the RNA, so that ribosomes attachment ensures protein formation is going to start over the surface of the RER. We even learned that there is something called as genomic RNAs, and genomic RNAs are found only in the case of the viruses. So there are certain viruses which do not have DNA but have RNA as their genetic material. So such an RNA is called as genomic RNA, and it can be double-stranded, example reovirus. It can be single-stranded, for example tobacco mosaic virus or TMV. So now we are left with the three main types, which are the rRNA, mRNA and the tRNA. We started a little bit about the rRNA in the last class. So let's start now. The rRNA is also called as the ribosomal RNA, and we learned in the last class that it is 70 to 80% of the total amount of RNA. So when we consider in a eukaryotic or a prokaryotic cell the total amount of RNA, then out of the total amount 70 to 80% of the amount is constituted by the rRNA. That means it forms the bulk of the, or it is the most abundant type of RNA. Then we learned that there are 3 or 4 types of rRNA depending upon whether it is a prokaryotic system or the eukaryotic system. So if it is a prokaryotic system, then there are 3 types of rRNA possible, and according to their S values they are 23S, 16S and 5S. And in the case of the eukaryotes there are 4 types of rRNA possible: 28S, 18S, 5S and 5.8S. So the 5S is the common to both prokaryotes and eukaryotes, and this is the proof, as one of the proof or evidence for the course of evolution, that we all have evolved from the similar machinery. That's why prokaryotes and eukaryotes both carry 5S type of rRNA.